For decades, the standard biological model has depicted the human cell as a fluid-filled sac, a loose balloon of cytoplasm where floating chemicals occasionally bump into each other. That model is obsolete. The cell is actually a tensegrity architecture. Tensegrity refers to a structural system where rigid struts, microtubules, are held in place by a continuous network of tension-bearing elastic cables, known as F-actin. Historical molecular biology viewed cellular signaling strictly through the lens of soluble biochemistry. The assumption was that information traveled exclusively via passive diffusion, relying on a ligand, a signaling molecule, eventually finding its target receptor. In reality, because of its tensegrity architecture, the cell exists in a constant state of mechanical prestress. The entire internal structure is pulled taut, holding its own shape against external pressures, and remaining physically poised to react immediately to applied forces. Ignoring this physical architecture obscures the actual ultra-fast mechanisms that govern cellular homeostasis, which is the state of steady internal conditions maintained by living systems. The cell is an active mechanical entity. Chemical diffusion is a slow, random walk. Molecules drift through the cytoplasm, taking time to trigger local reactions. Mechanical signaling operates on an entirely different timeline, relying on the instantaneous propagation of physical forces across a fully connected cytoskeletal network. When physical tension strikes the membrane on one side of the cell, that force ripples instantly through the cytoskeleton directly to the nucleus. This physical transmission delivers information at the speed of a vibrating string, vastly outperforming chemical ligands. This concept establishes the core logic of mechanobiology. Extracellular physical cues, such as the stiffness of surrounding tissue or the shear stress of moving fluids, directly dictate the cell's internal genetic expression. Diseases like Alzheimer's, Parkinson's, and ALS are widely studied as biochemical anomalies. However, the evidence points to a collapse of this exact mechanical transmission system. The physical architecture of the neuron fails. To understand how the brain degrades, we must map the precise pathways a healthy cell uses to translate physical push and pull into biological instructions. The process begins at the boundary. Mechanosensing is the cell membrane's ability to perceive external physical cues through proteins that physically change their shape when placed under stress. The primary mechanosensors are the mechanically activated, or MA, ion channels. The piezo-1 and piezo-2 protein families are the critical operators here, translating membrane tension directly into electrochemical signals. This diagram shows piezo channel regulation. Externally, mechanical forces like membrane tension or collagen 4 stretch the lipid bilayer, pulling the channel open to let calcium ions flood in. Internally, proteins like filament A desensitize the channel, forcing it closed despite outside tension. The cell membrane acts as an active mechanical receptor. It converts raw physical force into an electrochemical signal, initiating the next phase of the process. Once the signal is inside, the cell executes mechanotransduction. This is the journey of converting localized mechanical stress into biochemical signals that travel inward to alter gene expression at the nucleus. This chart maps mechanotransduction pathways. External mechanical stimuli, mediated by integrins, inhibit the internal beta-catenin destruction complex. Freed from the membrane, beta-catenin physically shuttles directly into the nucleus, acting as a transcriptional coactivator to turn target genes on or off. Structural tension physically relocates critical proteins from the outer boundary of the cell into the nucleus. This bypasses the need for traditional chemical triggers. The dominant mechanical signaling route regulating cell survival is the Hippo pathway, and its transcriptional coactivators YAP and TAZ. This is the master regulator of mechanical response. Extracellular matrix stiffness drives cytoskeletal tension, physically inactivating the core hippokinases. This allows unphosphorylated YAP and TAZ to enter the nucleus, promoting survival. If tension drops due to soft matrix or crowding, the hippocore activates, destroying YAP and TAZ. The physical rigidity of the cell's environment is the ultimate arbiter of its genetic destiny. It controls core programs for survival and differentiation purely through structural tension. In neurodegenerative diseases, the threat originates inside the cell. The internal architecture itself becomes the source of pathology. The process begins with a biochemical error. Mutant proteins misfold and begin to self-assemble within the neuron's cytoplasm. This triggers a biophysical mechanism called liquid-liquid phase separation, a process where a mixed solution separates into distinct phases. The misfolded proteins condense out of the fluid cytoplasm, hardening into dense, solid intracellular inclusions. A vicious cycle begins. These solid, rigid aggregates physically collide with the surrounding F-actin and microtubule networks, destabilizing the cell's internal load-bearing cables. This loss of cytoskeletal integrity removes the spatial regulation normally provided by structural proteins like vimentin. Vimentin is an intermediate filament responsible for organizing damaged proteins. Without its containment, the pathological inclusions accumulate rapidly. The cell's internal tensegrity architecture is shattered. Crucial survival signals like YAP and TAZ are trapped in the cytoplasm, effectively blinding the cell to its own structural collapse. The downstream victims of this mechanical collapse are the cellular organelles. Organelles rely on precise cytoskeletal tension. Consider mitochondria. Normal tension regulates division, but as the tensegrity network fails, pathological F-actin polymerizes around endoplasmic reticulum contact points. This actin physically chokes the mitochondria, inducing aberrant constriction and lethal fission. The Golgi apparatus also fails. The loss of integrin-mediated adhesion and overall cellular tension triggers Golgi fragmentation. This destroys its trafficking function through the disruption of ARF1, a key protein that regulates vesicular transport. At the endoplasmic reticulum, pathological membrane tension forces an abnormal localization of the piezo-1 channels. This structural error forces the channels open, triggering an unregulated release of calcium into the cell. Highly localized biochemical aggregates cause systemic cell death because they physically sever the load-bearing mechanical networks that connect these disparate organelles. Compare a healthy tensegrity network, accurately transmitting stiffness signals to the nucleus, against the diseased state, where solid aggregates have severed the lines of communication. Neurodegeneration originates from a total failure in the crosstalk between mechanical force and biochemical signaling, surpassing the simple accumulation of mutant proteins. The future of neurodegenerative therapy must look beyond simply clearing misfolded proteins. Research must pivot toward targeting mechanosensing channels, like piezo and TREC, and mechanotransduction pathways, like YAP and TAZ. To resolve diseases like Alzheimer's, Parkinson's, and ALS, interventions must repair and restore the structural biophysics and the tensegrity of the brain's cellular architecture.